All right. So hi, everyone. Welcome to the weekly cancer seminar. I have the privilege of introducing today's guest speaker, Dr. Catherine Flores. Dr. Flores received her PhD in hematology, stem cell biology from the prestigious Imperial College London in the United Kingdom. She did her postdoc at Duke University, where she trained with Doctors Philip Sampson and Dwayne Mitchell, two prominent figures in the field of brain cancer immunotherapy. She is currently an assistant professor in the brain tumor immunotherapy program at the University of Florida, Gainesville, where she focuses on developing cell-based immunotherapies to treat both pediatric and adult brain cancers. Dr. Flores is the recipient of 2 R01 grants and a multi-PR on a U01 grant, all of which focuses on developing immunotherapies for central nervous system derived cancers. She has published in highly regarded journals such as Clinical Cancer Research, Science Advances, and Nature Communications, and her work has led to several patent applications for cell therapies. Impressively, she's designed and conducted preclinical studies that fed directly into 3 of the first ever human clinical trials to evaluate adoptive cell therapy to treat pediatric recurrent medulloblastoma, high-grade glioma, and diffuse intrinsic pontine glioma, all of which has resulted in FDA IND approval. Currently, Dr. Flores is working to increase the efficacy of immunotherapeutic platforms using combinatorial hematopoietic stem and progenitor cells. So thank you, Dr. Flores, for taking the time to meet with us and share with us your recent work. I'd also like to extend some thanks to the faculty members and students who have met and will be meeting with Dr. Flores throughout the day. And as a reminder, if there are any questions, please leave them in the chat and Dr. Flores will address them after her presentation ends. Dr. Flores, the floor is yours.
Thank you so much for that very kind introduction, Phoebe. Thank you so much for inviting me to be here with you today virtually and giving me the opportunity to talk about the work that we do down in Florida. Let me share my screen. All right. Can you see the screen okay?
Yes. Yep.
Excellent. So as Phoebe mentioned, my primary focus is on the development of cell-based immunotherapy against both pediatric and adult CNS malignancies. My disclosures. So just a super brief background on brain tumors. So brain tumors are the most lethal cancer in children to date. They've surpassed leukemia as the most lethal cancer in children. Glioblastoma and high-grade gliomas are the most common primary brain tumor in adults, and it's also the most aggressive brain cancer with very grim prognosis. Median survival, overall survival is just barely over a year, 13 months, with a 5-year overall survival rate of less than 6% as of 2017. Although there are many ongoing clinical trials that try to target high-grade gliomas right now, a systemic review published in Neuro-Oncology last year stated that the 3 with the highest median survival against glioblastoma, only bevacizumab extends survival to 18.2 months. Optune, which uses Tumor Treating Fields, you may have seen the commercial. They've got they're the little like a little mini helmet or skull cap that emit energy that target the tumors, only extends median survival by about 20 months. And peptide vaccines conducted in trials at UCLA have only extended survivals by 19 months. So there's really a lot of work to do in this space. This is an example of survival curve of patients 70 and over. So frequent clinical practice includes biopsy for brain cancers and, if possible, tumor debulking. So those that can only have a biopsy, if the tumors are in eloquent tissue or in inoperable. So often we'll get a biopsy, but as you can see, their median survival is very low. And even with the bulking in this older patient population, the median survival is still below 12 months. It's about 9 months. So what I really want to talk about today is how we can overcome the major challenges for efficient therapeutic development against CNS malignancies. Because, I was just talking about how challenging and terrible this disease is. So now hopefully I can show you how we can help develop better therapies by overcoming these major challenges. So one is addressing tumor heterogeneity of brain tumors, specifically gliomas. So they're very heterogeneous tumors, both on the cellular level and antigenic level. Second, a huge hurdle is overcoming the immunosuppressive tumor microenvironment. With the cancers being in the brain, they're very silent and cold tumors. We don't want a whole lot of inflammation in the brain because it's your brain. And two, more and more data is coming out that really we need to start looking and treating at brain cancers as systemic diseases and look at peripheral immune modulation that brain tumors do to host immunity. So the tumor microenvironment is both very heterogeneous and also very immune modulatory. So these are the main, so in this caricature, let me get a pointer. These are the major cell types found in high-grade glioma. So first here in A, we have macrophages, microglia, and myeloid-derived suppressor cells, MDSCs, that are recruited from the periphery by the tumor cells themselves. And these recruited cells then in turn feed the tumor cells with cytokines that promote tumor growth. So TGF-beta, EGF, IL-6, IL-1 beta. They promote tumor growth, but also shut down anti-tumor immunity. And then here in B, looking at dendritic cells. So there's been recently a larger body of work demonstrating that dendritic cells in the at the tumor site, in the tumor microenvironment, are necessary and required for immunological rejection of tumors. And this, and more data is coming out that this is also so in brain tumors. Neutrophils, large amounts of neutrophils are recruited into intracranial tumors and circulating. They're both found in circulation and within the tumor. And neutrophilia is often used as an indicator to drug resistance in the high-grade tumors, not so much the low-grade tumors, but more in the high-grade gliomas. And here in D, lymphocytes. Lymphocytes are my favorite cells. So lymphocytes play many roles in cancer. So in an intracranial glioma that's been left untreated is you can find CD4 T helper cells, small populations of cytotoxic CD8 T cells, but largely we'll find T regulatory cells, which shut down any sort of anti-tumor immunity provided by the other lymphocytes. And lastly, more recent data has shown that astrocytes, host-derived astrocytes, actually do play a role and regulate cell survival pathways in cancers, so which is really interesting. And so those are the other cells that make up a brain tumor. But then looking at the tumor cells themselves, so this very elegant study used single cell RNA sequencing in combination with other molecular methods to further understand intratumoral heterogeneity the two most common primary brain tumors. So glioblastoma in adults here up at the top, and medulloblastoma in pediatrics. So they have identified 4 transcriptional cellular states in GBM and 3 subtype specific metaprograms in medulloblastoma that really underlie the intertumoral heterogeneity in these 2 brain tumors. And then at any given point, they found that each of those cells is actually in transition between cellular processes and differentiation states. So that means they're at different points of the cell cycle, different stages of differentiation. And then that also means that they're antigenically expressing different antigens at different levels. So then it really makes treating brain tumors kind of like, I like to think of, one of those air pillows. So you squish one end and then the other end expands. So like with brain tumors, we can, specifically target some cells, but then the other cells are still free to expand the population. So, just a little bit challenging. So to overcome this, we've developed two of our main platforms here in our program, which I'll go into more detail later. Our adoptive cellular therapy, which is more of a personalized medicine approach, really, which relies on ex vivo activated anti-tumor T cells. And also another platform, which is a little more broad than personalized medicine, is combining PD-1 checkpoint inhibition with a hematopoietic stem cell transfer to manipulate the tumor microenvironment. And this platform's been efficacious in multiple genetically distinct models of, or orthotopic models of brain cancers. So high-grade glioma here in KR158B, patch medulloblastoma, and brainstem glioma, which harbors the M27M mutation. So now I'm going to kind of explain how we address each of the major hurdles in efficacious development of therapy with each of our platforms. So adoptive cellular therapy, it takes total tumor RNA. So we take the entire tissue of the tumor. So since brain tumors are largely heterogeneous, we're taking all of it. So all the RNA, normal, neoantigen, pulsing it into dendritic cells and using dendritic cells to expand autologous tumor reactive cytotoxic T cells ex vivo with low dose IL-2 to expand tumor reactive T cells. And then that's adoptively transferred into the host after host conditioning. So either radiation or chemotherapy in case of the human. And that's followed with a hematopoietic stem cell transfer, also autologous, to manipulate the tumor microenvironment. And then maintenance immunity is maintained with dendritic cell vaccines. So this platform has been, and kind of iterations of this platform have been developed into clinical trials for children with recurrent disease. So children with recurrent medulloblastoma PNATs. And we've recently opened 2 trials, one evaluating this platform in children with high-grade gliomas, and then another in children with brainstem gliomas. And so also in our murine models of this platform has shown significant efficacy. So here in our orthotopic high-grade glioma model, KR158B, K2 brainstem glioma, and then our NSC group 3 medulloblastoma. So the point being that this cellular therapy can kind of surpass the different genetic drivers of the different tumor types, and so hopefully bypasses the tumor heterogeneity. So the bulk really of this platform are the T cells. So that's probably although all components are necessary, this is probably the most important part. So we expand a polyclonal population of tumor-specific T cells. But within this population, it's actually only about 5 to 7% of these T cells can actually recognize the tumor. But through TCR sequencing and TCR V-beta spectrotyping, just through flow cytometry, we can actually identify the tumor-specific T cells, the tumor-specific V-beta family. And to do this, so to generate the T cells, we take tumor cells, isolate the RNA, pulse them into dendritic cells, and use them to expand splenocytes in mice or peripheral blood in humans. And then in both, we get a polyclonal population of T cells. So we've got all these T cells. So how do we figure out which ones are important? So you can just back sort out the different TCR beta families by flow, and then use them as effectors against the tumor cell targets. So here, this example shows T cells against the NSC medulloblastoma. So we can see here in green is our positive control, that it's these TCR beta 13 and 8.1, 8.2 that secrete interferon gamma in response to recognition of the tumor cells. So then we can look in vivo in C57 Black 6 mice treated with adoptive cellular therapy. We can measure, looking at the spleens, and the peripheral blood, we can measure relative expansion of the different beta families and significant expansion of different V-beta families in responders versus non-responders. So in the non-responders, we kind of have a baseline, and I don't show it here, and I apologize, but it's just kind of like a baseline relative expansion of the different V-beta families, and they're all about the same. But in responders to therapy, we see these spikes. And depending on the tumor type we use, we see different spikes of different V-beta families, which are predictive of response. We can then take the spleens of these responders, sort them back out into the different effector populations by TCR V-beta family, and then target them against the primary tumor cells. So here again, we see TCR V-beta 8.1, 8.2 family and V-beta 13 family showing recognition of the primary medulloblastoma tumor cells. Since we're a translational program, we're always kind of trying to see if we can find some sort of biomarker of response, especially against brain tumors, because you can't, it's a little bit challenging to just go back into the tumor to look for response or to image the tumor. So here in mice that we treated with adoptive cellular therapy, we took the peripheral blood at 30, 50, and 120 days post-therapy. So these were responders. And here looking at the pie graph, we see we measured relative frequency of the TCR V-beta family expansions. And we see here in blue that the V-beta 13 family, which we had previously shown is tumor reactive in vitro and in vivo, that family actually shows expansion in peripheral blood 120 days post-treatment. And then looking at that family, looking at that family at the clonal level, we took the top 1,000 most productive, most frequent clones using TCR sequencing, and saw that here each color line is a single clone. But here we see that this blue, this clone expanded to up to 6% of the T cell repertoire found in peripheral blood. So as point of reference, the chances of running into any other different T cell clone is less than 0.01%. So 6% is huge. So then to prove that, so we said, okay, that's wonderful and everything, but are those really the cells responsible for rejection of the tumor? So with this medulloblastoma, we can measure tumor growth in the cerebellum using bioluminescent imaging. Here in this graph, here in black, we've got the responders to therapy, and in the red were the non-responders to therapy. We're able to measure peripheral blood over time. So we tag the tumor reactive T cells that we transferred in with DS red, so we can kind of track those cells longitudinally. And so what we found was we could associate a expansion of TCRV beta 13 T cells in the periphery with the mice that responded to therapy. And here is an example of one of the controls of TCRV beta 4 expansion. So this V beta family did not expand in the peripheral blood, whereas these guys did. So we then did another experiment where we adoptively transferred only the V beta 13 T cells, and then our control bulk unsorted tumor reactive T cells, and adoptively transferred those into the orthotopic tumor bearing mice. And we found no significant difference in survival between these V beta 13 T cells and the unsorted T cells, which tells us two things. Is one, we successfully identified the V beta T cell family responsible for rejection of this specific tumor. But two, since there's no significant difference between that and our bulk unsorted T cells, from the translational standpoint, we can get away with potentially just adoptively transferring the unsorted T cells, but then also that also suggests that since only 5 to 10% of these unsorted T cells are actually tumor reactive, we need to figure out what the other T cells are as well. So we looked at the same platform in children, in a child who was long-term responder. So she had a recurrent medulloblastoma, received adoptive cell therapy, high-dose chemotherapy, followed by autologous peripheral blood stem cell transplant, followed by autologous tumor-reactive T cells, and also then followed with 3 autologous vaccines, pulsed with her own tumor RNA. So again, we did TCR sequencing and looked at her peripheral blood 2, 6, and 16 weeks post-therapy. And we found, looking at the pie graphs, each of these is a single clone. So about 25%, a quarter of her, the T cells in her blood comprised of only 5 different T cell clones. And looking at those cells, so indicating that, you know, we tried to make the association that we see the clonal expansion, we see a response. So we're hoping to accrue more patients so that we can actually make that firm association, but hopefully we can do that very shortly. And looking at these 5 clones, each of those belong to different TCRV beta family. And each of those families largely comprise of a single expanded clone. And so we were able to take just a commercially available antibody against, here we chose TCRV beta 9, sorted those cells out of some of her cells that were collected 16-week post-adoptive cell therapy here, and used them as effectors against target cells, which were her own dendritic cells pulsed with the original tumor RNA. So there were autologous cells presenting antigen from the tumor, and then measured interferon gamma expression. So here we see that that TCRV beta 9 family T cells were able to recognize cognate tumor antigen presented by the dendritic cells. So we were able to expand and identify the tumor reactive T cells without needing to know exactly which antigens are being expressed or being targeted. Because as you see here, right, we see just in this pile alone, we see at least 5 different families and clones that are expanding and being targeted and targeting at least 5 different antigens in the tumor. So here we're bypassing the tumor heterogeneity of the brain tumors. So with this adoptive cell therapy platform, we can generate tumor reactive polyclonal cytotoxic T cells in both human and mice. We can identify the TCR beta families, which are tumor specific, and potentially use a clonal expansion of the tumor reactive T cells in peripheral blood as a potential biomarker of response. And then clonal expansion may be predictive of response, which is really a huge benchmark for immunotherapy in this patient population. So next, we want to address the tumor microenvironment. So in both of our platforms, we leverage a hematopoietic stem cell transfer to help manipulate the tumor microenvironment to sensitize it to either the adoptive T cell therapy platform or the checkpoint inhibitor. And I will show you some evidence that that is occurring. So in our tumor microenvironment, here's our tumor microenvironment. So in an untreated, so this data is in orthotopic high-grade glioma, the KR158 model. So in untreated mice, we see a large majority of the bulk of the tumor is actually MDSCs and TAMs. But when we look at late-stage tumor that's treated with either adoptive cell therapy with HSC transfer or PD-1 plus HSC transfer, we see significant reduction in MDSCs at the tumor site and significant reduction of TAMs at the tumor site. And here is an example experiment from adoptive cell therapy. So looking at MDSCs, which we're marking here with CD11B and GR1 per milligram of tumor, we see a large amount of MDSCs, but that's significantly reduced with just the T cell, adoptive transfer of just the T cells and the dendritic cells. But when we add a stem cell transfer, that is even further reduced. So the frequency of MDSCs are reduced with the immunotherapy, but even more so with stem cell transfer through mechanisms which we don't quite understand yet, but we are trying to elucidate. So looking at the dendritic cells. So one of the goals with brain tumors is to get dendritic cells to the brain tumor. And dendritic cell vaccines have been around for a very long time, but those act more peripherally in draining lymph nodes rather than at the tumor site themselves. So what we've done, so hematopoietic stem cells differentiate into your lymphocytes, your myeloid cells. They differentiate into MDSCs, but also dendritic cells. And now I live in the South. And there's a Southern saying that goes, you are who your friends are. So if hematopoietic stem cells are around like immunosuppressive MDSCs, they're receiving those cytokines and are driven down that pathway. But when you expose them to interferon gamma, TNF alpha, GM-CSF, they're driven more down a dendritic cell pathway. So when we combine these HSCs with immunotherapy, let's see, again, we see a decrease in MDSCs. But then we see an upregulation of CDC1s in both adoptive cell therapy platform and the checkpoint inhibitor, and an increase in CDC2s. So the CDC1s, we believe, are the ones responsible for in situ T cell activation, and the CDC2s, we believe, are the ones that migrate, that leave the tumor. We think they get to the tumor, grab some tumor antigen, and then migrate to peripheral lymph nodes to activate host T cells. And then the HSC-derived cells at the tumor site upregulate markers of dendritic cells, so CD11C, MHC class 2, CD886. And then looking at the lymphocytes, so there's been a lot of literature prior to us showing that bone marrow derived stem cells, both hematopoietic and mesenchymal, actually migrate to solid tumors and also migrate to high-grade gliomas via the CXCR4 SDF1 axis. So what we have found is that when we adoptively transfer hematopoietic stem cells with tumor-reactive T cells, so the stem cells get to the brain tumor, but then they bring the T cells with them. So they increase accumulation of tumor-reactive T cells at the tumor site. And those T cells are largely interferon gamma secreting CD8 T cells. So here with our adoptive cell therapy, in an untreated glioma, we have very little CD8 T cells. But then when we, let's see, 5-gray is 5-gray irradiation. So when we irradiate and add T cells plus dendritic cells, we see significant increase in interferon gamma secreting CD8 T cells at the tumor. But then when we add the hematopoietic stem cell transfer, that further increases the accumulation of the CD8, cytotoxic CD8 T cells at the tumor site. So we're increasing interferon gamma secreting T cells. We're also, and we have some pilot data showing that we can also reduce Tregs at the tumor site through mechanisms we don't quite yet understand. And in our checkpoint platform, when we combine PD-1 with hematopoietic stem cell transfer, we see the same trend. So here we have what are called great mice, and great mice express YFP on the interferon gamma promoter. So our hosts are great mice, and they were treated with either no treatment, stem cells only, checkpoint only, or the combination hematopoietic stem cells plus anti-PD-1. And then these late stage tumors were treated. We then excised the tumors and looked for YFP expression on the CD3 T cells, and we see significant increases in CD3 YFP positive T cells in the tumors that got the combination therapy. So we see here increases of marker of T cells. So this is, we also see increases in markers of T cell activation. So CTLA4, interferon gamma, fast ligand, TNF, but we also see a decrease in markers of suppression at the tumor site with our combination therapy, which is really huge because we can target TGF-beta, IDO1, INOS, PDL1, all at the same time. And therapeutically, using single drugs, that would be too toxic to do with drugs and inhibitors in mice, and I'm sure in humans. We also see in the combination therapy, we also see increases in CD8 T cells in the spleens of treated mice. So to summarize this section, so what we think, so we know hematopoietic stem cells migrate to the tumor. They secrete MIP-1 alpha and recruit more T cells from the periphery, both host T cells and adoptively transferred T cells. These T cells, then these tumor reactive T cells can then recognize and kill tumor. thereby secreting interferon gamma. And so interferon gamma actually drives hematopoietic stem cell differentiation into dendritic cells. And we think this happens in situ at the tumor site. And then these hematopoietic stem cell derived dendritic cells can then subsequently enhance the T cell activation that we're observing, which we think contributes to overcoming treatment resistance to checkpoint blockade alone, which we are also observing. Does anyone have any questions? Sorry, I tend to speak quickly on Zoom. Okay. So even though we're seeing responses, we are also seeing the majority of the mice in our tumor curves still succumb to tumor. So we have done some studies to try to get at mechanisms of escape from immunotherapy. So here again, we have our orthotopic intracranial high-grade glioma. Here it is untreated, and here it is treated with adoptive cellular therapy. But then we see that, the mice still succumb to disease. So what are the escape mechanisms? So we isolated these isolated tumors from the mice that succumb to tumor, which escaped from therapy, and did some simple tumorigenicity assays. Here's an example of one. So the mice that escaped adoptive cellular therapy are significantly more aggressive than the primary tumor here in the solid line, which is expected. These tumors underwent a large amount of immune editing and have learned and have been trained to become very aggressive. So we just then did very simple bulk RNA-seq. We took, here's TOGA 1.1, it's the one that got away. And this was the first one. So TOGA 1.1 versus the parental KAR158B line, and just did simple bulk RNA-seq, and saw that they were genetically different from each other. So they differ in the immune-related pathways, and more importantly, the cell cycle, division, chemotaxis pathways. So we know that the escape tumors are genetically distinct, but how that translates into therapy, we're still unsure. We then wanted to know if they're immunologically distinct, because if they were not, if they were immunologically similar, could we just go in with a second wave of those T cells? But these experiments suggest that we cannot. So back to our adoptive cell therapy platform, we generated KAR158B specific T cells. So T cells that target the primary tumor, and then generated the TOGA 1.1 T cells, which are T cells that specifically target the escape tumor. And then use those T cells as effectors against the primary cells, the KAR158B, B16 as a control, and then TOGA 1.1, the escape tumor. And what we found was the primary cells does have some recognition of the TOGA 1.1 of the escape tumor, but not very much. The amount of interferon gamma secreted was very low. But here, when we look at the TOGA 1.1 T cells, so this T cells that can specifically target the escaped tumors, has also very limited recognition of the primary tumor. Although they can see each other, the response is not as robust as the actual tumor they're generated against. So kind of digging back to our toolbox to be able to identify which TCRV beta families are responsible for tumor rejection. Here in the KAR158B, so we wanted to see if they were the same families or if they were distinct families suggesting that they are completely antigenically different after escape. So here, running our assay against the primary tumor, we see that the T cells expressing TCR-V beta 5152, V beta 6, and V beta 8182 are the ones that recognize the primary tumor. But looking at our escaped tumor, here we see that they both share that the TCR-V beta 5152 family can recognize both tumors. But here it's the TCR-V beta 8 family that can recognize the TOGA, so the escaped tumor, but not the primary tumor. And same here, the V beta 6 and V beta 8182 T cells can recognize the primary tumor, but not so much the escaped tumor. And as you can see, we're expanding multiple polyclonal T cells that are reactive to the tumors. So then we asked, okay, well, that's great, but we then asked if the escaped tumors are similar to each other, because that might suggest a secondary method of therapy, but they are actually very distinct from each other, which poses yet another challenge for recurrence. So here we generated TOGA 1.1 T cells against TOGA 1.1 and TOGA 1.2, which came from a different mouse that escaped the same primary therapy. So these TOGA 1.1 T cells can recognize TOGA 1.1, but that recognition is significantly decreased of the TOGA 1.2 tumor cells. And here, looking at TOGA 1.2 tumor cells, these cells do not recognize the TOGA 1.1 tumor cells at all. So there's no recognition of the other escaped tumor whatsoever. And again, analysis of this shows here TOGA 1.2 cells. It's the TCR against this escaped tumor. It's the TCR-V beta 3 and the TCR-V beta 7 families that are reactive against this tumor. So this is highly suggestive that we are definitely seeing antigen escape variants as an escape mechanism from adoptive cellular therapy. But we also looked at other global mechanisms of escape. So the ultimate goal is to try to figure out, okay, if people are escaping our therapy, what else can we give them to either prevent escape, or what can we give them after they escape or they recur? So here we looked at MHC class 1. So here's our KR158B primary tumor, looking at MHC1 expression. And here our TOGA escape tumor cells actually downregulate MHC class 1. So and untreated tumors have high amounts of MHC class 1, and that significantly decreases after the mice receive adoptive cell therapy. And in our TOGA 1.1, so our tumors that escape adoptive therapy in the first place have lower amounts of MHC class 1, but then that's when we give them second adoptive cell therapy with a TOGA 1.1 T cells, that MHC class 1 is reduced even further by both relative frequency and MFI. So we have antigen escape variants and reduction of MHC class 1. And what we also found was that adoptive cell therapy increases the amount of PD-L1 expression by the tumor cells, both by percentage and by relative expression by PCR. And what's interesting is we find a correlation between PD-L1 expression and MHC1 expression in the tumor cells after escape. So since there's such high amounts of PD-L1, we did what was pretty, it begged to be combined, adoptive cell therapy begged to be combined with anti-PD-1 checkpoint inhibition, which we did. So what we found here, back to our orthotopic high-grade glioma murine model, we combined adoptive cellular therapy, here shown in the dotted line. In a solid line, we combined adoptive cell therapy with anti-PD-1 and had significant increases in overall survival in that primary tumor. However, in the tumor that had escaped, we generated, this is a total 1.1 tumor, we generated adoptive cell therapy using TOGA-specific T cells, and then combined that with PD-1 inhibition, but we saw no significant increases in survival. So that suggests that in the recurrent setting, which is more aggressive, checkpoint inhibition with adoptive cell therapy may not actually be a good plan for overcoming tumor escape. So here in our, so I hope I showed you that with the tumor escape, our TOGA cell tumor lines, let's see, we have antigen escape variants as a source of escape, downregulation of MHC1, and upregulation of immune checkpoints. And so we expect to see multiple mechanisms of escape and, possibly redundancies in the pathways that lead to escape. But what we were hoping was to be able to kind of overcome that and either prevent escape or identify how to go in with a second treatment after recurrence. So that those mechanisms are still underway. And so lastly, just in the last couple of minutes, the current directions of the lab right now are really to look at peripheral immunity and to see how we can leverage peripheral immunity to our advantage to make to kind of switch peripheral immunity to be more pro-tumorigenic than anti-tumorigenic. So there are many studies in other solid peripheral tumors that show that cancers essentially hijack the immune system. So we have expanded MDSCs and neutrophils. Let's see, there's increased levels of GM-CSF and IL-6 in blood serum, which promote MDSC development. In the spleen, there's dysregulated extramedullary myelopoiesis. And in the bone marrow, people have shown that bone marrow-derived cells can promote vascularization and angiogenesis of tumors. And in glioblastoma, the group out of Duke has shown that T cells are actually sequestered in the bone marrow. So we kind of took a step back. So we've been looking at the, at the immune cells, but so now we started looking at where the immune cells come from. So they come from hematopoietic stem and progenitor cells in the bone marrow. So in the murine system, we've been using lineage negative cells as our HSPC compartment, so hematopoietic stem and progenitor cells. So then we just wanted to see what the impact of brain tumor was on hematopoiesis. So we did just bulk RNA-seq to compare lineage negative HSPCs from healthy non-tumor bearing mice versus glioma bearing mice. And we saw that they're genetically different, but they're also genetically different in important pathways such as homeostasis, IL-4 and IL-13 signaling, extracellular matrix organization. And there were 133 significantly upregulated genes by glioma-bearing hematopoietic stem cells and 302 significantly downregulated genes in stem cells from tumor-bearing mice. And we also have some pilot data showing that hematopoietic stem cells are actually epigenetically dysregulated by just having an intracranial tumor. So we did a simple characterization of the progenitor cells in the bone marrow, and we find that the bone marrow of tumor-bearing mice have increased common myeloid precursors, increased granulocyte monocyte precursors, increased subsequent MDSCs, increased monocyte dendritic cell precursors, and increased macrophages, all in the bone marrow. So really, the bone marrow is reprogrammed to be more immunosuppressive. We've also started to analyze peripheral blood from GBM patients, and we're seeing similar patterns. So increases in GMPs and increase in CMPs. But since humans are, in no way uniform, we still need to accumulate significant amounts of peripheral blood from newly diagnosed patients to be able to conclusively make these arguments. So far, what we've seen is that gliomas can manipulate hematopoietic stem and progenitor cells at the genetic level, skew relative frequencies of the myeloid compartments, and also alter sulfate differentiation. And ultimately, we are actually working on being able to determine sulfate differentiation and drive sulfate differentiation away from an MDSC phenotype and more to a dendritic cell phenotype to promote anti-tumor immunity. So hopefully I've demonstrated how we can overcome some of the major challenges in therapeutic development for brain tumors. So overcoming tumor heterogeneity by adoptive cell therapy, overcoming immunosuppression in the tumor microenvironment with the hematopoietic stem cell transfer. We've identified some immune escape mechanisms and are currently also working on looking at peripheral immune modulation in tumor-bearing hosts. So this work is largely, the data you saw today is largely generated by the two postdocs in my lab, Bailey Dean and Tyler Wilds. I think Tyler is actually trying to do PATH in Colorado. I'm not sure. And then my two grad students, Connor Francis and Laura Falsetto, and this is done in collaboration with the Immunotherapy program at the University of Florida. Thank you very much. I know I spoke a little bit quickly, so I'm happy to answer any questions.
All right. Do we have any questions from the group? Catherine, wonderful presentation. It's great data. I mean, I recall visiting you guys some time ago, and I think, the lab was getting set up and you just made tremendous progress. Look, just, really great.
Thank you. Thank you so much.
So I guess as people are kind of, digesting some of this and preparing questions, I do have one question. So there was a specific TCRV beta family member that, at least in the mouse model, seemed to predominate and then expand over the course of several days or months, I think maybe. So a couple of questions. One is, aside from the fact that it has, that specific TCR, have you interrogated perhaps at the genetic level what might be imparting that T cell with enhanced persistence and killing activity?
We have not yet. No, we haven't. We have not yet. I think actually Dr. Mitchell is trying to do exactly what you said by TCR sequencing and single cell seq in patients who have responded and are showing response and showing also expansion of specific T cell clones.
Okay, and then one more question. I think we can go on to Jill's and Sid's questions. Have you identified the antigen that TCR recognizes?
No, it's on my list. It's on my list, but like that's everyone's first question, like what the antigen is. In the murine system, we have not. I mean, that would be easy to do. it would be more challenging in the humans. So in the humans, I showed example one patient, but we're actually seeing different, and the hope was that we would see expansion of the same clone with all the kids, but they're actually been very different. So we've kind of we're stepping back to kind of figure out a way to identify those antigens in the humans. I mean, that would be gold.
Yeah. I mean, that's really the what everyone's after these days. We might not find a common antigen per se, and that would be what it is. Jill, do you have a question?
Yep. Hi, very nice, very fun. I also love antigen questions. So I'm wondering though, you have a number of different clones. Do you think they all see different antigens? Have you tried putting them through any of the programs, like the Mark Davis type programs, where you can see if they may recognize similar structures?
So we have started to do that in collaboration with another PI at the university. So far, no. So far, they don't see similar antigens, but just trying to find the alpha beta, pair is also something that we're trying to figure out in the murine system. And so the way a program works is I do a lot of the murine analysis, and there's another group in our program who does a lot of, parallel human analysis. And so really there's more effort in the human side trying to find the alpha beta pair, but then also trying to see if the different clones recognize the same structures. So far they don't recognize the same antigens, but I don't know if they kind of on a structure epitope level, you're not sure if they're the same. So you mean like crystallography?
Yeah, there's programs that will just predict cross-reactivity.
Oh, I see. I'm sorry, I misunderstood the question.
So I wonder if you look in certain V beta family members, if those might, I mean, there's just so many cells that react. I wonder if those would be cross-reactive with the same antigens.
No, I have not checked. Do you have a favorite program?
Well, I haven't tried it myself, but I'll give you details. I meet with you at 3.
Oh, yes.
Okay.
Yeah. Great. Thank you, Jill. Hey, Sid, you had a question as well?
Yes, and it will be away from antigens because they scare me. One question I had was that I'm especially very interested in this added response that you see from the PD-1 treatment after ACT. And I mean, the real question is, normally we see very little PD-L1 expression. In this model, is there more expression, or does the PD-L1 expression also change after your ACT or HSC treatment?
Would you mind? I'm sorry, you were like breaking up. Would you mind repeating the question again?
Oh, sorry. So my question is, normally we see very low amounts of expression of PD-L1 in the tumors, in especially human tumors. That's why I get interested in this PD or additional added PD-1 response that you see. And do you change expression of PD-L1 in the tumor after HSC or ACT treatment? Why do you think you end up getting this increased response when you see no response without them?
So interesting question. So after adoptive cellular therapy, we do see an increase in PD-L1 expression at the tumor site.
Oh,
Um,
okay.
yeah, but not, so several things increase the PD-L1 we find. So irradiation will increase it. And then the T cells themselves, when we do just a T cell transfer, we see upregulation of PD-L1 as well. So that's why we decided, well, okay, let's see if it works.
Actually, I had another question. I'm going to sneak it in here. This is, I don't know if you remember Peter Fecci's work in T-cell sequestration in the bone marrow. And so do you see that? Do you see any sort of interaction of those increased amounts of CMPs and monocytes with the sequestered T cells in the bone marrow? I mean, I think that bone marrow story is really exciting. And
Yeah. No, the bone marrow, yes, no, the bone marrow story is very exciting. So we do see, so it's interesting, we see that sequestration of the CD4 T cells in the bone marrow of certain, when the mice have certain tumors. So like E0771, when they have the intracranial breast, like fake met, LLC1, fake met, and then some of the brain tumors, but not this KR158B high-grade glioma. So there's something definitely tumor specific that leads to the shifting in the bone marrow population, which I think is really cool.
And so do your ACTs end up homing into the bone marrow in those mice? Like, do you see sort of, I mean, that could be one of your quote unquote, drop in efficacy because something is happening that's sort of attracting them into the bone marrow instead.
We do actually see those T cells in the bone marrow as well, but I've never checked to see if they're being stuck there. That's a great idea, Sid. Thank you.
Okay, so that will be the last question. We do have a meeting with the students in the same chat. So all the students that are meeting with Dr. Flores, we should just stay here.
Excellent. Catherine, we'll catch up later. Thank you.
Hey, thank you so much.
Nice job, Catherine. See you around.
Bye, Sid. Thank you so much.